Hello, everyone. Conrad from the past here. By this point, I'm sure you've had just about enough of listening to Conrad from the future and his bad puns. So please enjoy my delightfully short and pun-free presentation on a component of my master's research in which I examined the drivers of regional scale variability in the abundance of an invasive bryozoan in the kelp beds of the Northwest Atlantic. And this project was co-supervised by Dr. Anna Metaxas and Dr. Danielle Denley. So the Northwest Atlantic, for the purposes of my study, is defined as the area shown in this map here in eastern Canada and the United States. And in this region, much of the subtidal rocky bottom is covered in aggregations or beds of kelp, which are these large habitat-forming macroalgae shown in the photo. In the Northwest Atlantic, these kelp beds consist of three numerically dominant species listed here, the specifics of which I don't really have time to go into, but all of which are shown in the photo here so you can see what they look like. Now, we care about these kelp beds because they support biodiversity, commercially fished species, and kelp aquaculture. Now, about 30 years ago, an invasive bryozoan species was introduced called Membranipara membranacea that began colonizing kelps in the Northwest Atlantic. Now, this bryozoan has an annual life cycle where it begins as planktonic larvae, which settle out of the water column and onto kelps in the spring. And these settlers, which are the white dots on this kelp here, then asexually reproduce to form large colonies throughout the summer, which in turn weaken the kelp blades and ultimately lead to the loss of the kelp as we get into the autumn. Now, most of the membranipara colonies on the remaining kelp then die off over winter, and the whole process starts again the next year. But the adult abundance of membranipara, which we measure as the percentage of the kelp covered by the bryozoan, is highly variable between different sites and from year to year, and so is this resulting kelp defoliation. So what's driving this variability? Well, past studies have identified multiple variables that may affect the percent cover of membranipara, and these are temperature, kelp substrate, which means the species of kelp on which the bryozoan is growing, water depth, wave exposure, and the density of kelp at a given site. However, past studies producing these findings have had a limited temporal and/or geographic range, so it's unclear if these findings are generalizable to the entire Northwest Atlantic in space and time. Secondly, it's also unclear how important these variables are in determining percent cover, both relative to one another and, in the case of some of them, whether or not they affect percent cover at all in an absolute sense. So the objective of my study was to address these knowledge gaps by quantifying the relationships and determining the importance of these candidate predictors in determining the percent cover of membranipara in the Northwest Atlantic. And to do this, I used a statistical meta-analytic approach, which I'll go over briefly for you now. So to begin my analysis, I compiled a large data set of observations of percent cover and the candidate predictor variables from throughout the Northwest Atlantic region, from a combination of historical data shown in the blue and newly collected data by me and my lab group, which are the orange points on the map here. Once I collected all my data, I used a model averaging framework based on linear mixed models to calculate parameter estimates for each predictor, from which I could assess the importance of each variable and its relationship with percent cover of membranipara. And here are the results of my analysis, which it turns out mostly align with those of previous studies. So I'm showing variable importance here in the black circles, and then the relationship between each predictor and percent cover of membranipara in the white circles. So temperature was the most important predictor of percent cover and had a positive relationship. Kelp substrate was the second most important predictor, where the cover on one species of kelp, Laminaria digitata, was greater than on the other two. And then depth was the third ranked predictor and had a negative relationship with percent cover, which is actually just due to the decrease in temperature with depth because I had used sea surface temperature. And then finally, wave exposure and kelp density did not affect percent cover at all. So in terms of implications, with temperature being the major driver of percent cover, we can expect warmer waters due to climate change to lead to increased percent cover of membranipara in the Northwest Atlantic. However, my results indicate that this increase will depend on the species of kelp on which the bryozoan is living and the depth in the water column. But regardless, future increases in percent cover mean we will likely see further losses of kelp, meaning it's all the more important to protect the kelp that we do have left in our region. And to this end, I've begun work on a species distribution model for percent cover in the Northwest Atlantic, which I can use to estimate the current and future distribution of member nipper cover to inform kelp conservation. And with that, I'll finish by acknowledging all the people and funding that made this work possible. Thanks for listening, and I'll be happy to take any questions. Back to you, Conrad from the future.